So just pardon if I sneeze a little bit. I see that the target and usual the pill and fish don't mix. To you further in my talk, what does this mean? Probably read about these news. Chemicals in your furniture can mimic hormones in your body. Also, pesticide can cause frogs to change sex. And very recently, household dust can make us fat. That's probably why I'm gaining weight uncontrollably now. So maybe we should always clean our house so that we can lose weight. And finally, this is old news, but I think this is very important. Pollutants can change sex in fish. What are these things? What does this mean? These unusual things that are happening to our body and to other organisms to the presence of an array of endocrine disrupting chemicals. They can be naturally occurring compounds, metals. So what are endocrine disrupting chemicals? Before these chemicals, let me define first endocrine system, which system that is targeted by these endocrine disrupting chemicals. So what is an endocrine system? It is the system of the body consisting of our glands that controls the functions of cells and tissues in our body. And these endocrine disrupting chemicals, we can abbreviate it as EDCs, are substances or chemical compounds that can alter functions of the endocrine system and consequently cause adverse effects in organisms. So they can either mimic naturally occurring hormones in our body. That means they're gonna have similar effects, or they can block the receptors in our body that binds these hormones functions in the body. Other impacts or effects of these endocrine disrupting chemicals or EDCs in human and wildlife includes reduced sperm count in humans, eggshell thinning in birds, reduced male sex organ in reproductive function in seals, and development of male sex organs in female sea snails. Already mentioned earlier, feminism of male fish. So their impact on life, EDCs can also affect the economy. How they can affect the economy? EDCs can affect growth and consequently population of fish, which can adversely affect nature. When eaten, these species exposed fish can pose health risks to humans, consequently affect fisheries and aquaculture. These health risks either indirectly brought by EDCs cost the US $340 annually for healthcare. So how to take these chemicals? This can readily occur through the gills, through the diet, to the skin or through maternal transfer. In the case of maternal transfer, that accumulated in the mother's egg can be transferred to the developing embryo by absorption or consumption of the for embryo development. Fish are very sensitive to these chemicals. From various sources may accumulate in coastal water. Some of these EDCs can mimic, which is, or the estradiol 17 beta, which is the main female sex hormone. These chemicals, such as or EE2, which can be found in birth control pills, which has, with, as I've mentioned, with the main female sex hormone beta, and the donelphenol or NP, which can be found in agricultural and even in household products, can affect growth and reproduction. So in order to understand and to analyze the effects of these chemicals in warm water species, there are a lot of studies already in temperate species of cold water species or fish known in warm water fish. So in order to determine the effects of these chemicals in the growth and reproduction of warm water species or tropical fish, we use the Mozambique tilapia. Mozambique tilapia is a type of tilapia native to South Africa. So why Mozambique tilapia? Tilapia because they can grow and spawn in captivity, very resilient. They're very tough fish. They're warm water species. They're euryhalion. That means that they can tolerate a wide range of salinities. In coastal waters and wetlands or rivers. Thus, they can be a good modeling the effects of these endocrine disrupting chemicals in warm water fish. Further, let me give you a brief background of growth in fish. I just made it as simple, so pardon me. So in fish, growth hormone is a hormone responsible for the growth, is secreted from the pituitary, which is for the base, located at the base of the brain. So this growth hormone binds to its receptor, which is called growth hormone receptor in the liver, then stimulates the liver to produce insulin-like growth factor. This insulin growth factor, or IGF, binds to IGF-BPs, or insulin-like growth factor binding proteins, as the carrier proteins in the bloodstream. Then this IGF binds to its receptor in the target tissue, or the growth hormone itself can directly bind to its receptor in the target tissue in fish. With regards to reproduction, which is also secreted from the pituitary gland, stimulates or activates both male and female reproductive organs development. In female fish, it activates the follicle cell, which is the cell responsible for hormone secretion in the ovary or the female reproductive organ. The follicle cells, by the action of gonadotropin, produces 17, which, as I have mentioned earlier, is the main female sex hormone. With the help of its receptor called estrogen receptor induces or activates egg cell development. Stage of egg cell, which we're gonna call vitellogenic phase. This is the phase eggs accumulates vitellogenin, which is the main precursor of made the egg yolk protein. So during this stage, secretes E2, which is the main female sex hormone. This E2 stimulates the liver to produce vitellogenin. This vitellogenin then accumulates in the ovary, which acts as the major precursor for egg. Egg yolk protein is important for the nourishment of the embryo. Thus, this protein and receptor could be a good indicator to analyze the effects on reproduction in fish. Our preliminary data in adult tilapia showed that of E2 at 5 microgram per gram body weight and NP at 150 microgram per gram decreases expression such as the growth hormone receptor or GHR and the insulin-like growth factor or IGF, but they increase plasma vitellogenin. This implies that NP can mimic the action of the natural E2 and thus can affect growth and reproduction in adult tilapia. The long-term effects of early exposure to EE2 and NP. Hence, in order to determine the long-term developmental effects of EE2 and NP, we expose yolk sac fry of tilapia. Yolk sac fry are just the baby fish with the egg yolk protein still attached to their body. So this is our experimental design. So you can see here the yolk sac fry. We collected the yolk after they already consume all the yolk sac, and for 21 days to E2 at E 0.1 to 1 microgram per liter and NP at 10 to 100 microgram per liter. After exposure to either fresh water or seawater, and then grow them for 3 months. After 3 months exposure, plasma, muscle, gonad, or reproductive organ, and pituitary will be sampled. The experiment is already ongoing. This photo shows our experimental setup. So these are headers which contains water with chemicals that supplies doused with EDCs to the containers with the fry or baby fish. And this shows the yolk sac fry at the start of experiment. Shows the fish 2 months after the end of exposure, so you can see they already grow bigger. After 3 months post-exposure, the effects of these chemicals on the parameters of growth, as I've mentioned earlier, the growth hormone, its receptor, IGF, IGF-BPs, and for reproduction, vitellogenin, its receptor, and the ratio of gonad weight, body weight, and sex ratio. So I would like to thank my supervisor, Seal, and our collaborators and associate, Drs. Lerner, Professors Moriyama, and Reeves for their support, and Mr. Cody Petrosacoma and Mr. Austin McPherson for their help.
Aloha, good evening. My name is Leilei Shih. Go ahead. Okay. I'm a PhD student, and today I am going about an invasive species called MYCALE grandis. This invasive sponge is doing to affect the ecology on coral reefs here in Hawaii. So as I mentioned about MYCALE grandis, this sponge, is that it's invasive, meaning Hawaii on ships, it's not native to Hawaii. So it arrived as biofouling. And what's important about this is that this sponge was able to voyage halfway across the Pacific from a completely different location, halfway across the Pacific Ocean through different temperature gradients, and then settle in a completely new location. So it means that it's very adaptable and can settle in an entirely new environment. This sponge MYCALE grandis you see is on the right, bright orange encrusting sponge, and it's squishy and it overgrows degraded corals. That's how it settles. It's an opportunistic sponge. We have coral that are away either through pollution or climate change. It gives an opportunity for this invasive sponge to take over. My study site is in Hanauma Bay, which you can see on the left, and this is on the windward side of Oahu. So what exactly is a sponge? Well, it's an animal, it's not a plant. It's actually the first multi-organism to have evolved on Earth, and it's 600 million years ago. What about that is that it has survived several of Earth's major mass extinctions. Some of the same extinctions that killed out all kinds of other species. Again, a resiliency and adaptation. Today, there are over 8,000 known species of sponges, but we believe that there are a dozen species of sponges, and they exist in oceans all across the planet, in all kinds of environments, depths, and habitats. And sponges, especially in the tropics like here and in the deep ocean, can live to over 200 years. In fact, just 2 years ago, scientists came across an enormous sponge in the deep waters off Hawaii, and this sponge can actually be one of the oldest animals on Earth. It's centuries to millennia old, years old. It is the size of a minivan. It's about 12 feet across. You can see this is a underwater vehicle here. It's 12 feet across and 5 feet high, and this was discovered in about feet of water. And it was actually discovered in Papahanaumokuakea, which is, for now, at least a national monument. But MYCALE grandis, the sponge that I studied, isn't only an invasive sponge, but these are some of the other well-known ones. What makes MYCALE grandis interesting? Why are we more interested in studying this sponge than the others? It's actually the most widespread and most common invasive. So while these other sponges do exist, this is the one that we're keeping an eye on because it established itself pretty widespreadly in different environments. And in fact, just this year with the Smithsonian Marine Geology, they actually found 150 previously unseen 150 species of unseen sponges in Hawaii. A third of them, which are completely brand new species that we had not known anything about. Is really exciting. It's actually a collaboration between 24 different institutions. But what is that my research on the most widespread invasive sponge is going to give us a lot of context to understand how sponges, and in particular invasive effect that they have on our coral reef ecosystems. So one of the first things I had to do is look at abundances of MYCALE grandis in Kaneohe Bay. So it's found all throughout Kaneohe Bay, but it's in this region here. And this is likely due to the fact that there are higher nutrients, in particular organic matter, in the South Bay. And I compared my results to previous data that we already had on abundances Bay, previously done by Bishop Museum. And between 2004 and 2006, there was double the abundance on, and since 2006, returned more to about the same levels that we had in 2004. But this is still really high for a sponge invasive. What this is indicative of is that the sponge has established itself on the reef. I forgot to mention earlier that the sponge was actually only first seen in 1996, so it's actually a pretty recent invasive species. All right, well, we're ready for this slide. So why is MYCALE grandis so important? So sponges are full of microbes. They have a very diverse and community, kind of like the ones that we have in our gut, even more concentrated and more active than the microbes that we have in our gut. And hence, they're often referred to as microbial fermenters. In fact, 40% of the volume in sponges. I shared my research here. I talked a lot about this process that sponges perform called nitrification. And since I talked about it last time, I don't want to get into it this time. But to let you know is that this process that the sponge does perform actively is very important to the story that I'm telling you today. So basically, sponges are able to convert one form of nitrogen into another form of nitrogen, and they do this very actively. This is very important for ecosystems because, as you may be familiar with, nutrient levels of coral reefs. Nutrients, you can get algal overgrowth, you can get something called eutrophication, and this overgrowth of algae in coral reefs is bad for coral reef. What's important about this ability to transfer to nitrogen into another is that it's changing the nutrient levels on reefs and hence able to affect overall ecosystem function. Is so the other important thing about sponges is that they vary seawater through their bodies. This is how they feed. And this is a video that I made of MYCALE grandis pumping fluorescein dye. So I injected this fluorescein dye along the, and you can see these jets. The sponge is actively pumping. There are about 5 jets on this sponge. So as you can see, it's very prolifically pumping seawater. And this is important because, as I mentioned, they are chemically the nutrient profile on coral reefs, and they're actively pumping the seawater. So they're exposing all of this seawater to their microbes and changing the nutrient level in the seawater. I've measured MYCALE to be able to pump 100 times its own volume of seawater a day. And this is a big deal in Kaneohe Bay because in the South Bay, where this where I'm studying this sponge, sticks around for a very long time very quickly. So basically, the sponge has the ability to come into contact with and truly affect the nutrient levels in the seawater there. Sponges pump seawater using specialized cells called choanocytes, and this is what they look like here. These special cells have a funnel and a flagellum inside the collar that rotates like this, and it beats seawater to pump it, and it does this very efficiently. So not only do they have these specialized cells to pump seawater, they are actually very clever, and they've taken advantage of physics to help them pump even more seawater through their body plans. And they use something called Bernoulli's principle that you know from high school physics. But what Bernoulli's principle states is that when you move from a smaller volume into a larger volume, there's going to be a drop in pressure because you're going from a smaller space into a bigger space. In fluid dynamics, the velocity or the speed of the water will have to speed up because you're going from a smaller space into a larger space. So sponges have actually incorporated their body plan in order to take advantage of Bernoulli's principle and to do some of this work of pumping seawater for free. So they're very clever animals. Now, the last thing that I want to talk about today, new research that I'm presenting, stable isotopes to tell us something about what sponges eat and to figure out what their trophic level is. And what that means is we want to figure out where they are on the food chain or what position they have in the ecosystem. So for example, plants and algae are at the base of the food chain, and they have a very low, and top predators like sharks are at the top of the food chain, so they have a very high trophic level. And the isotopic composition of tell you where in that spectrum it is. And it isn't just levels 1, 2, 3, 4, 5. It can be anywhere in that spectrum, 7, 3.4, for example. So it tells us very specifically where something. So I'm actually still waiting back on some flow cytometry data that will tell me about the exact microbes that I have in my sponge. So I can't give you an exact number today, but I can tell you that when I was preparing sampling, I separated the microbial community that I told you about from the sponge tissue itself. And I looked at these two things separately. And what I did see is that the sponge has a higher trophic level bacteria do. And obviously this makes sense. So we know that the sponge is actually consuming some of its bacteria, and it's consuming some of the detritus that it's pulling out of the seawater when it's pumping. So we do see some information in the relative difference between the isotopic data. So this is sort of a not a very familiar topic, so I wanted to draw and to explain it via what you eat. So fruitcake versus marshmallows. So I'm normally comparing heavier isotopes to lighter isotopes. So you can think of the fruitcake as heavier isotopes and marshmallows as lighter isotopes. And when you eat fruitcake, it's very heavy. You're going to feel like SpongeBob here. And when you eat marshmallows, it's very light. You're going to here. And the heavier to be retained in you, it's going to get incorporated into your tissue because it's not as easy to get rid of it. And the lighter food or the lighter isotope gets excreted and goes away. And so what happens is your tissue is actually recording everything that you ate. It's creating a unique isotopic signature about what you ate. And why this is an important tool is in the past when we were curious about what something ate, say we wanted to know what a fish was eating, what we would do is we would cut open its stomach and we would look at its gut contents. And then whatever was in there, we would say, this is what the fish eats. But that made it true. I don't want to be morbid, but say like you just had a sandwich for lunch, and if we cut you open and looked at what was in your stomach and we saw a ham and cheese sandwich, we shouldn't conclude that you only eat ham and cheese sandwiches. Sometimes you might eat salad, but we only know what you last ate. What's powerful about stable isotopes is that it can tell us all of the things that you eat and specifically in what proportion you eat these different things. So this is what stable isotopes can tell us. So what are some ongoing applications of this research? It's very important to study invasive species on coral reefs because it helps us create policy for coral reef conservation. As I mentioned, this sponge is able to actively influence seawater chemistry and can shape the ecology and community structure on the reefs. And also, native species and invasive species is a very important issue in Hawaii. We actually have more endemic species in Hawaii than we have anywhere else in the world because of our unique geographic location. We're more geographically isolated than anywhere else on Earth, and therefore we have more unique endemic species than anywhere else, and that's why Hawaii is so special. But that makes us very vulnerable to invasive species. So studying invasive species like this sponge will help us with conservation efforts and state management on invasive species issues. So I wanted to just say thank you to Hawaii Sea Grant for their support and these various facilities that I was able to conduct my research in.
All right, everybody staying warm out there? You jump around a little bit. Oh, we're just gonna, not ready for that yet. Okay, my name's Derek Kraft. I'm a PhD candidate under Dr. Brian Bowen at the Hawaii Institute of Marine Biology. And I'm really lucky to work with a nice guy named Richard Coleman, who's my lab mate, and him and I work together on a project called Fish Flow, and we look at where fish populations are coming from and where they are going. So as you're all aware, we're at the beautiful Hanauma Bay. Did anybody go in the bay today? You did? What'd you guys see? A lot of stuff? Say fish. Fish, all good. I'm glad you saw fish. Yep, it's a fish talk. So even though Hanauma Bay, it's been protected since 1967. Hanauma Bay has these thriving fish populations, and what most people don't realize is these fish populations that are here very much rely on other parts of the ocean to replenish them. And just as Hanauma Bay relies on other parts of the ocean rely on Hanauma Bay. And these connections are really important. And where that is driven from is the life cycle of reef fish. They start, well, there are adults here, and there's two fish. They just said they like each other. They spawn, so they shoot their eggs and gametes into the water column, and they get fertilized. And now you just have this little floating egg, and this egg can just drift off with the current down the coast or wherever it's taken until it develops into this larva and beats it as fast as it can to the nearest reef where it settles. Becomes a recruit, starts to grow up, becomes an adult, and the cycle continues. However, this lower part of their life cycle here, they have very, very small home ranges. I would even say like a square kilometer is pretty big for a reef fish. That dispersal and population connectivity relies on this planktonic stage, or when they're just little eggs. And turns out those little eggs are really hard to track and know how far they go. To some advances in molecular techniques, aka genetics, that. And here's a little example. So Mark Christie and his team went over to Hawaii, and were interested in looking yellow tangs. And these guys are really important for the aquarium fish trade. Populations weren't doing so well, so they went and looked at these populations, and what they were really interested in was where they were spawning and then where they were recruiting. So they went around and they collected a bunch of adults and a bunch of juvenile fin clips, non-lethally when they could, and they used a parentage analysis, which is kind of like how you would identify who the parent is, an illegitimate child, kind of along those lines. And what they found was that down here in the south part of the island, where they matched some adults to some of the juveniles down the coast. So they said, okay, well, there's 6 areas, and then being recruited down here are really important. Aquarium trade collectors collect the fish down here. The populations up the coast will be affected, and they'll be reduced. They'll get less recruitment the next year. So yeah, these things were important for the aquarium trade, but we all know that they're important for other reasons, especially for people to eat. So reef fish were a big part of what people ate around here. Currently, still 31% of Hawaiians residents fish for them to eat, to feed their families, to feed their, and about 30% of the total catch is attributed to those recreational fisheries here on Hawaii and in the other islands. This fishing is fairly unregulated. There's been quite a shift in the ecosystems around here, and a lot of that can be attributed to overfishing. What I'd like to do is highlight a connectivity between spawning sites and recruitment sites, some areas that are really crucial, where they're spawning more and maybe where they're recruiting, and eventually that'll lead us where these fish are coming from and where they are going. So some Hawaiian community leaders got together. They gave us a list of fish that they were concerned about, and we kind of whittled that down to about 5 species we thought of samples and represent a good diversity of different types of reef fish. So they're the square spotted goatfish, the yellow convict hang, the gold ring surgeonfish, the bluefin trevally. There we are. I'll link those areas of spawning to those areas of recruitment with multiple species, and we're going to look across the entire Hawaiian archipelago. Just like the study that Mark Christie did, we're going to collect adults and juveniles from all these areas. Then we're going to extract the DNA, run it through a high throughput sequencer, and then we're going to compare the adults and the juveniles and hopefully find some of those parent-offspring connections, and therefore drawing lines between these different sites. Where do we do that? Okay, so this is Kaneohe. It's located up here. We're in Hanauma Bay, so it's kind of just up around the corner. All of these little black dots represent little patch reefs. So this is an example of connectivity on a really on the right shows residence time of the water. So the northern part of the bay up here, this water is exchanged every 1 to 5 days. We're down in the southern part of the bay, every 25 to 30 days. So the water isn't moving nearly as much down here. The model to demonstrate this is still Kaneohe Bay. And you're going to see these little colorful dots start moving around. Now say those are different parents. They're spawned into the water column, and now they're set adrift. So notice the blue and green down in the southern part of the bay, and the yellow up in the northern part of the bay where the water's exchanged more. They start to, the red and the yellow start to move towards the mouth of the bay, and out they go. Into the open water they go. And what you can see, now what's mostly left is the blue and the green. Fairly predictable body of water. We can say that maybe the fish down in the southern part of the bay are seeding the northern part of the bay. And then when those fish spawn, it's going out, and we don't know how far it's going. So we're going to zoom out a little bit. Here's Kaneohe Bay again this time, but what I want you to notice is what's happening outside of the bay. How much more the water is moving and how much more those different colored eggs are being distributed. So once you're out into larger bodies of water, this becomes much harder to predict. It will be as much. It's fairly chaotic out here. So again, the eggs can leave the bay. We're not sure how far they're going, maybe up to Laie, maybe they make it over to Haleiwa, or maybe they make it all the way over to Kauai. So not only are we interested in the fish in Kaneohe Bay and around Oahu, we're also interested, sorry about that, Kaneohe Bay, like a small part of Oahu, but Oahu is a small part of the whole Hawaiian archipelago. So what we've done is we've sampled a large number of fish in the bay. We've sampled over 1,000 fish around Oahu and about 1,500 total across the Hawaiian archipelago. We went over to Kauai earlier this year, collected on the north and south side, as well as Maui, collected on the north. And over on the island of Hawaii, we actually organized a team of marine biology undergraduate students at Hilo, and they spent a couple months traveling around the Big Island collecting fish for us, and we're very thankful for all the samples. So what we've got so far, 100 samples have been collected. All of the DNA has been extracted, it's been sequenced. We just started getting back information a couple weeks ago, and just this week we've been analyzing it, so we're really, really excited about that, and I'm super sorry it's not ready for this talk today. We hoped it would be, but it was not. So we're looking forward to actually getting some results back. But what we can maybe expect, like model, it's really hard to predict where those eggs might go. But what we do know is that they will travel along the current. So we do expect fish to the west to east direction, along the current. And additionally, what we see in Alaska is genetic breaks here between Oahu and Kauai, mostly due to the distance. And then also between the Big Island and some really strong currents pulling across here. So to see as many eggs traveling across these gaps. Like I was saying, these results can be really important as far as fisheries management goes. If we find some areas are much more crucial, that's what's creating fish further down these islands or other islands. These set up fishing regulated areas, so we could show that these are working, or we could possibly add more. And then also overall connectivity between Oahu and the other islands, and how other islands also affect Oahu. And then we can take these that we've studied thus far and hopefully make judgments about similar species and get a good idea of what's going on in multiple species. So most of what we're looking at, as highlighted here, is the connectivity between spawning sites. So next time you're out snorkeling in Hanauma Bay or somewhere else you hold dear to your heart and you see a cute little reef fish, you can look at where are your parents and how far have you come to get away from them. So with a project this big and with this many samples, we have a lot of people who come out and help us collect. Richard, he's been this entire operation. Our other lab mate, Michael Hoban's been very involved. Our advisors, Brian Bowen and Rob Tonin, Sea Grant, and the Castle Foundation for funding, and a ton of field help. And with that, I think we'll all stand up and take your guys' questions. Yes.
I have a question for our first speaker. It's a question I've asked. We talked on this subject in the last 5 years. One end of the spectrum, natural estrogen getting in water, and then artificial human use, and then you have and chemicals in the plastic. Which is the worst?
So it depends, because EE2, the one in birth control pills, has, for example, estrogen, the natural female hormone. Like, for example, we gonna assign 1. The effect is 10 times if the same concentration. And NP, I think, is 100 times lower. So
That's the chemical itself. Now, the life of the chemical in the water, plastic, stay around forever.
yes.
And
So
ingested by all kinds of critters.
yeah.
But it's dissipated.
So these chemicals, the EE2 and which is the ethyl estradiol in birth control phenol, they accumulate and they're persistent. So in Hawaii, we don't have any data yet on the levels of these chemicals, but in the mainland, they have some data where we based the concentration that we use for this experiment. So they are based on the concentrations that are found on the mainland. Thank you.
Oh, yeah. Oh, that was, so, I mean, that's not even a sponge that's in Hawaii. Sponge and muted, that's in the Florida Keys. But I just wanted to put in a playful image of the sponge. Oh, yeah. It doesn't, I mean, as long as you don't actually, or die. Yeah, the person survived, and hopefully this one survived too.
It found until if sponge is plastic.
Oh, yeah. That's a really good question. That's not one that I thought of. I think that we could find. So I don't think that the sponge. So it's actually selective feeder. Have these amoebocyte cells that go, so when it pumps the water and it's filtering all the seawater through its body and it catches all these particles, the amoebocytes, the specialized cells that can actually travel all throughout the body of the plant, they're fascinating creatures, I'm telling you, can actually go and they pick out specific food particles and then that they want and they incorporate it into the sponge. So it's not even bacteria that they want to eat or all the plankton that it wants to eat. So I don't think that it will ingest plastic on purpose, but as they've found plastic in fish tissue, I think we could probably, there's any plastics in sponges. It's not a part of my research, but is seeing that we've found plastic in coral and we've found plastic in fish and in sediment and whatnot. So we wouldn't probably do it through isotopes because that's looking at the isotopic signature in biological material. So this is actually something that sponge researchers as an entire community are constantly trying to figure out. And sponges in different ecosystems, I think, have different needs. So some sponges have photosynthesizing bacteria and they can get carbon through that source, and others are eating other forms of heterotrophic bacteria, so synthesizing bacteria. Some are actually just taking out better or different like pieces of nutrients, if you will, from the water. And this is kind of what we're trying to figure out and when they need it. So I don't have a specific answer for you, but this is the question that we're. Oh, yeah. I would, it's probably, but again, they do have these, so, I mean, different from that's actually getting fed bits of plastic from its mom, these specialized cells are actually going and selecting exactly what they want. And when I was talking about how sponges harbor these entire microbial communities within their bodies, there are some researchers that are looking at are sponges actually bacteria for their own need. So sometimes there's this relationship, this symbiotic relationship between the microbes and the sponges, and where the sponges are providing a habitat, a safe space for the bacteria that's safer than the water. But at the same time, cultivating it for its own use, and when it's ready, it eats its own bacteria too. Okay.
I know what releases, but how long do the eggs get to stay in the nest?
Yeah, that's a great question. How long are the eggs in the pelagic stage? It can be anywhere from 3 weeks to a couple months. Yeah, and it's very species. I know some.
And specific, let's say, let's talk about yellow tan. Are you drift around for weeks and then settle?
Yeah, absolutely. I mean, the farthest one one of the longest dotted lines was 114 kilometers, very south part of the island up to the north part of the island. And I mean, we know that a lot of the times they do make it from one island to another. And it depends where they are too. So some places that have really specific currents, if they're an island has a current that has a big loop, you'll find a lot of the fish there the larval stages. So when the current brings island, that's when they're turned into larvae and try to settle. So it's species specific and then it can also be location specific.
There's someone about sponges in the Caribbean. And tell me if I heard you wrong or what, but it seems like you said they used something about them in cancer.
Yes, so this whole field, but this again has to do with the microbial community that's within the sponges. And this is one of the most incredible things about sponges. So it's really not the sponge performing these different chemical reactions that I was talking about, changing one form of nitrogen to the other, but rather it's bacterial community. And these bacterial communities are making different chemical compounds. And in sponges, a lot of these compounds are defensive. And what that means is if a sponge wants to settle, because sponges settle too, they have a larval stage as well. It doesn't want to compete with other organisms, so it takes advantage of its bacterial community that has its ability to make these chemical compounds that can or deter other organisms from growing around it. It's an advantage. So that's an example of a chemical compound that a sponge can make. And these compounds are used a lot in drug discovery because these are obviously very powerful compounds. If it can kill other organisms, it could potentially kill cancer. And of the compounds that's used in treatment of leukemia was originally isolated from sponges. So I don't know anything beyond that because I'm not a medical doctor. But yes, yeah. So sponges are actually one of the is that people who do pharmaceutical and drug discovery research, they look to sponges just as they go to rainforests because there's just so much biodiversity and biological activity, and they're hoping they're my drug to find.
My question, I'll confuse us.
It was with the dispersal, with the yellow tanks, they find sort of from sort of modeling flow versus where the fish actually is?
I don't think so. There's not very many. Wasn't actually all that many parent offspring connections that they found. I think they found 4 of all the ones they collected. There was pretty small. So no, I mean these were all very concurrent with the currents that were going north. So I don't think they were surprised. We do actually have a whole portion of this project that other people are working on that's most where we got our little models from. So they're modeling on a bigger scale. I just don't have any of those to show you guys.
Funny. In your introduction, first slide, you went to those Reef Invaders, say a place and they come from the galaxy very far. And substrate, they make a formula that they have enemy.
Yes, so substrate is usually rocky substrate. They like something rocky, hard, and porous to attach to. So they settle other environments that coral, whether it's deep water, cold coral, or a tropical coral, likes to settle on. So they like rocky substrate. They do have some enemies. So some sea turtles eat sponges, and some fish feed on sponges, I think like parrotfish and whatnot. So I didn't mention the sponge that I study, MYCALE grandis, but previously it was unknown whether it or not it had any natural enemies or predators. And especially with invasive species in Hawaii, since it's not from here, there was no natural predator that evolved it. And for the longest time, we didn't know if anything wants to attack or eat MYCALE grandis. Now there's a postdoc at HIMB Jan, I forget. Yeah, yeah, there. So
You could be wrong.
okay, but I think that they're working on a cowries will eat the sponge, and they've fed the sponge to cowries, and the cowries will eat it, but they don't know if cowries on the reef go about eating the sponge, because if they're having it in an aquarium and that's all you feed it, the sponge, but if they're not that yummy, they might not eat the sponges in the wild. So that's the potential. This particular sponge in Kaneohe Bay. Friends, I would say that it's microbial communion because it's creating all these chemical compounds for them, it's providing energy, and it's providing food for them. They have some symbiotic relationships. And I don't think they really get a lot of benefit from any other organism other than the bacteria within them, but they do sometimes cohabitate with other organisms.